Hey, everybody. Hey, so for folks who don't know me, I'm Dave Haas. I'm the co-director of the Tennessee Center for AIDS Research. Our center, as folks hopefully know, is a four-way partnership between Vanderbilt, Meharry Medical College, Nashville Cares, and Tennessee Department of Health. And once a month, we have our Tennessee CFAR seminar series. So it is my great honor to introduce our speaker today, someone who has been close with me and a close colleague for over a decade now, and that is Dr. Bindong Liu. And so Bindong is a professor of microimmunology at Meharry, and he has been a co-director of our Tennessee CFAR's Laboratory Sciences Core for a decade now, since 2015. Which is soon to be renamed as our Advanced Technologies Core. He is expert in molecular virology. He has extensive experience in molecular and cell biology aspects of HIV, with lots of expertise in flow cytometry. In recent years, expert in CRISPR gene editing and various other HIV-related assays. His scientific focus has been on host restriction factors related to HIV, in particular ApoBec3G, and a host factor which restricts HIV and which HIV finds ways to circumvent through VIF. So much of Bindong's work is focused in this area. So I'm looking forward to hearing Bindong's talk. So with that, I'll hand things over to you, Bindong. I guess you have to share your slides. Great. Oh, and before we start, let me tell people, if you have questions or want to jump in, please raise your hands or use the Q&A function in Teams. It's in your menu, hopefully up top or somewhere on your screen, either the Q&A or hand raise, and we'll do our best to recognize you. And you can do that during the talk or at any point. Okay. And I should mention too, the director of our CFAR, John Curta, usually introduces these things, but he is on study section at the moment. He'll probably be joining us
during this. We actually just finished, so I'm here.
You jumped in. Okay,
but
so you reviewed your last grant in time. Good.
I have. There's a lot of good stuff out there. A lot of good stuff.
So all yours, Bindong.
Okay, thank you, David. So like David mentioned, I've been here like 2 decades, and David has been my mentor for like 2 decades. I really appreciate his advice, mentorship for my career development. So this is thank you for joining the seminar, and I really appreciate the opportunity to present our data. It's always exciting to talk about data. So let me get started. This is the title is a long title. I know it's a long title, but it's going to be easy for me to explain this during my presentation. So before jumping the data, I want to talk about a little bit about the epidemiology of HIV/AIDS, because I know in the audience we have some student, N1 student enrolled in our virology training program. The purpose for this slide is just to remember everybody, especially for the student, that we still have a big problem with HIV/AIDS, with approximately 39 million people worldwide live with AIDS, and 1.3 million people become newly infected almost every year. And more than 630 people died from AIDS-related illness. So basically, HIV/AIDS remains a significant global pandemic. The good thing is, luckily, we have a very good antiviral drug, even though we don't have a cure, we don't have a vaccine. The antiviral drug is very effective. Basically turn the deadly disease into a chronic disease. However, there are major issues later on with the comorbidity or everything, other things. One of the major challenges is drug resistance. So I have 2 examples over here. Those 2 drugs efavirenz, nevirapine, usually to be a major antiviral drug regimen using in clinical, but not recently anymore because the drug, of course, one of the reasons is the drug resistance evolved in those 12 countries. Once it reached a certain threshold, means the drug would not be able to use in the clinical anymore. So that gives us urgency for keep working on developing new antiviral drug. So that being said, looking for new antiviral drug development, and also one of the critical things is you need to have a new novel target for the drug development. So we have a very good target for a variety of things in terms of a viral component. Let's say we have a fusion inhibitor, we have an RT inhibitor, we have an integrase inhibitor, and we have a protease inhibitor as well. So those are all very mature, very good, potent antiviral drug. But we need to spend this. What if all those resources being exhausted and we do not have a new antiviral drug? Eventually, we will not be able to have a good research field. We keep thinking out of the box, try to find a novel antiviral drug target novel target for antiviral drug development. Throughout this presentation, I want to make the argument HIV-VAVE is a very good target. So VAVE is an accessory protein for HIV virus. Is essential for HIV virus replication. That means if something happened to the VAVE function, HIV virus will not be able to replicate anymore. So that's the unique part for this protein. It's unique. It does not have a human homologue. This is another important part because you do not want to use something who has a very close similarity to any human part. That will give you potential to generate adverse effect down the road when you develop antiviral drug. So those two are very unique feature for this protein, make it very attractive as a target for antiviral drug development. However, the last point I want to mention is the VAVE function is overcome a very potent antiviral host restriction factor. So with all those in mind, HIV-VAVE is a very attractive target. However, 2 decades has passed until we discover its function. We still do not have any candidate even close to clinical trial. So that tells us maybe something's still going on in terms of a molecular mechanism we do not fully understand, so that we still don't have a drug being developed. Of course, this is one of the reasons, and some other reason will not be the focus of for this presentation. So basically, I want to argue that we still have a lot of things we do not understand between VAVE and APRIL-BAG3D, or VAVE, HIV, and host interaction. So a little bit of background about APRIL-BAG3D. I mentioned this several times already. Is a host factor, is host innate immunity factor, which is very potent for HIV viral replication. Is deaminase enzyme. It belongs to APRIL-BAG family. There are a bunch of family members over here, but only in this presentation, I'm going to only focus on one of this A3D, APRIL-BAG3D, because it has the most potent antiviral activity over here. So it's been a classical idea how HIV-VAVE and APRIL-BAG3D interact each other. So this cartoon illustrated APRIL-BAG3D anti-HIV function. So I have a producer cell over here, means this cell produces virus. This is a target cell, means once the virus being produced, it will target to this cell, infect this cell. So during HIV viral production, Abrobag 3D get co-packaged into the viral particle. Upon infect the targeted cell, Abrobag 3D will reduce the viral genome cDNA production and also cause a, I think I should use a laser pointer. So this is a producer cell. This is the target cell. This is the virus being produced from this cell. And the Abrobag 3D got co-packaged into the viral particle. Upon infect the target cell, Abrobag 3D will do several things. The two major things is one is reduce the RT product, reduce HIV genome production. The second thing is induce G2A hypermutation in viral genome, may induce degradation or fatal mutation. So the picture over here right now is not very clear, but the general idea is it is how Abrobag 3D performs its anti-HIV viral function. HIV specifically develop a mechanism to overcome this by expressing an accessory protein called VAF, viral infectivity factor. HIV VAF actually induce Abrobag 3D for proteasomal degradation. Once this happens, there will be less chance for Abrobag 3D being packaged into the viral particle so that HIV virus can replicate. So this is one of the typical classical idea how HIV VAF overcome Abrobag 3D antiviral function. But over the years, evidence show that both clinical evidence and also laboratory evidence show that Abrobag 3D, even though being largely induced by HIV VAF for degradation, but there are still residual amount of Abrobag 3D being packaged into viral particle. So what does this residual amount of Abrobag 3D do to HIV virus, and whether VAF will still have effect to counteract this residual amount of Abrobag 3D, it was not very well defined. So basically, this is a major goal for this presentation for our experiment design and research. We want to find out after the degradation process, something incorporated into the viral particles such as VAF and maybe Abrobag A3D or VAF being co-packaged in the viral particle. What are those interactions further on? So this is the main theme for this line of study in our lab. And before doing this experiment, I want to mention two reports being published by two different groups. All of them report that 2 to 7 molecules of Abrobag 3D in the viral particle will be sufficient to completely inhibit HIV viral replication. So that's under the condition without functional VAF. And we all know with functional VAF, HIV virus replication totally fine. So this is those two reports give us a hint that maybe after the degradation, everything, VAF is still doing something to maintain HIV viral infectivity. So with this in mind, we perform a very simple experiment. So we want to see what will happen to two virus. One virus is a VAF deficient virus, means the virus does not have a functional VAF. Another one is a wild type virus, means it has a function. What will happen if we have an equal amount of Abrobag 3D being packaged into those two viruses? What about their infectivity? So by doing this, we produce virus using a transfection system. So this is just normal transfection what we usually do in the lab. VAF deficient virus, we use a small amount of Abrobag 3D because in this virus there is no VAF function. So Abrobag 3D will be packaged in the viral particle. Look at this band. This is a viral incorporation of A3D. In wild type virus, we have to introduce more Abrobag 3D expression to exaggerate wave function for its degradation. So we use a different amount of aprobax3D in the wild type virus, 12 micrograms, 16, 18, 20, that's a lot. The purpose is try to force aprobax3D into the viral particle. Then I want to draw your attention to those two viral sample. One is this one, another one is this one. You see the incorporated aprobax3D, this one a little bit more than this one. So in the wild type virus, we have a little bit more aprobax3D being packaged into the viral particle. And if you compare the viral infectivity, there are a huge difference. So over here, this virus, even though it incorporates a little bit more aprobax3D, its infectivity is much higher than this one. So that means because the virus difference over here is this one has a wave, this one does not. So that tells us wave may still doing something over here to protect viral infectivity. So this simple experiment gives us a confidence we move on to invest more effort on this project. So we want to introduce mutation. So that mutation resists to wave-induced degradation, so that the purpose is you want to have an equal amount of aprobax3D in the viral particle, so that you can compare the wave function, right? Otherwise, you would not be able to do that. So one of the mutant, which was discovered by other lab, is called aprobax3D D128K mutation. Basically, in the 128 residue in the aprobax3D protein introduced D2K mutation. Once the single mutation happened, HIV wave would not interact with wave A3D anymore. It would not inhibit aprobax3D and it would not induce its degradation anymore. This is a well-established, published data. So then we generate another mutation. So for wave to induce aprobax3D degradation, it is proteasomal degradation. And the proteasomal degradation, proteasome ubiquitin modification is essential. And the ubiquitin modification to the targeted protein happen in the lysine residue. Aprobax3D has about 20 lysines. We actually requested this mutation from other lab. They mutate all the lysine to arginine. Lysine, arginine, very similar amino acid residue. The only difference is arginine would not be able to modify it by ubiquitin. And we add HA tag to the N-terminal. Those are all potential targeted for ubiquitin modification. So the purpose for doing this is to prevent its polyubiquitination modification, so that the wave will not be able to degrade this. And we did this, and we achieved the goal that you see over here. So this is the two mutation. One is the lysine-free mutation, what I mentioned, we generated ourselves. This is D128K mutation, which was introduced by other lab. We co-transfected with 3 virus construct. One is wild type. This is a third wave mutation. This is a wave point mutation. I want to hold on this and talk about this later. Okay, so I want to draw your attention over here between those two samples. So this is a wild type virus. This is a wave mutant virus. And because this aprobax3D is resist to wave induce the degradation. So in the viral particle, we saw equal amount of aprobax3D being packaged into the viral particle. So then once we have equal amount of aprobax3D, then we look at their infectivity. You see this one. This one has wave. This one without wave. This is a log scale. You saw 10-fold difference. That means this wave, because the only difference between those two viral particles is this one has wave, this one without wave. We have equal amount of aprobax3D being packaged. So this means HIV wave may still protect HIV viral infectivity, tenfold difference. Okay, for another mutant, D128K mutant, because HIV wave does not interact with Abivax 3G D128K anymore, so it will not protect the infectivity. So you see wild type virus and the mutant virus all go down. This is a log difference compared with no A3G. This is 100-fold difference. Okay, so this data suggests HIV wave really still does something to protect HIV viral replication. Then we move on to look at the, remember I told you Abivax 3D induced G2A hypermutation. So with all those IC being set up, VARs being isolated, we want to see whether there's any effect, any difference in the G2A hypermutation rate. What we actually did was we harvest all those VARs, we infect sub-T1 cells in like around 20 hours or even 15 hours to 20 hours. We isolated cDNA. Then we PCR amplified the cDNA. Then we clone the cDNA into colonial vector. Then we do sequence to analyze G2A hypermutation rate over here. You see the G2A hypermutation between wild type VARs and the wave mutant VAR and the wave deleted VARs. There's significant difference. Means I got the opposite. So this is a wave deleted VARs. Wave deleted VARs generate a significantly higher G2A hypermutation compared to VARs with wave, functional wave. That's further again argue that HIV wave still does something over here to protect HIV viral genome. And once the wave does not interact with Abivax 3D anymore, this is the D128K mutation, equal amount of G2A hypermutation being generated between wave mutant VARs and wild type VARs. This is not. This is just a control. It's not a surprise to anyone. This is new. This is relatively new. So with that data in mind, I want to talk about something. Remember, we have a third VARs. The VARs it has a 3-point mutation in the C terminal of HIV wave. It was actually introduced before the discovery of a wave and Abivax 3D interaction by lab from Dana Gabus's lab, Harvard, Dana-Farber. So of course, before that, they do not know the interaction between wave and A3G. They found out some defect in this wave mutant, but they would not be able to correlate it with A3G. But that's not the focus for our experiment. We borrow this mutant. The purpose is this. So when we study the viral packaging, the wave viral packaging, so this is the cellular expression of wild type wave, B3 wave, and this is in the cellular expression. This is the viral incorporation. You see, this is a wild type wave. This is the B3 wave. B3 wave significantly reduces its viral packaging. And this is without A3G. This is with A3G and with A3G lysine free. The same thing happens, similar thing. This is a 50% compared with wild type. This is like 20% compared with wild type. So the key point is over here that the wave incorporation has a defect. Then we look at the viral infectivity over here. You see the viral packaging, all three virus, same amount of Abivax being packaged, but the infectivity over here is significantly different. This is without wave. This is a wild type wave. This is a wave with a defect, not 100% defect, some defect in viral packaging. You see the infectivity in between wild type and wave mutant. So we argue that for wave to perform this function, it has to be incorporated in the viral particle. It's in line what we talk about. This is the whole thing. The whole thing has to be has happened after the viral degradation, ACG degradation or everything. So it's happened after this wave. The wave has to be co-packaged into the system for wave to perform this function. Similar thing happened to G2A hypermutation rate. I'm going to give you some details over here. So basically, each dot represents one clone. So we analyze multiple colonies so that we can get a statistic significant result. So you see, this is a wild type. This is a wild type wave. This is a B3 wave. This is a without wave, and the hypermutation rate for B3 is in between wild type and wave mutation. So I further argue that wave really does something in this process to protect HIV viral replication. So this is the model we propose for this part. So for this is the part of a viral replication without wave. Without wave, ABRBAC3D being packaged, introduced into the target cell. ABRBAC3D will perform its hypermutation activity. The level of hypermutation reached to lethal level, so that ABRBAC3D would be able to make the virus uninfectious. Versus with wild type wave, HIV wave will somehow inhibit ABRBAC3D during this process, generate a small amount of a certain amount of, because it's balanced, generate a certain amount of ABRBAC3D and the leftover for the viral particle, because you look at the clinical sample, you always see the residual G2A hypermutation happen in the viral cycle. So one of the argument over here is the reason HIV would not 100% get rid of ABRBAC3D from its life cycle is because HIV want to take advantage of ABRBAC3D to introduce G2A hypermutation, to push forward its immunoadaptation or genomic variation to benefit its viral replication. We all know that almost all pathogen university take advantage of genomic variation to benefit its viral replication. So this is a summary for this part of our presentation. And the limitation for this whole set of assay is ABRBAC3D being overexpressed, because in a physiological setting, you never see that much of ABRBAC3D being incorporated into viral particle. So to study whether this is to study this question using more physiological relevant system, you have to use wild type virus. You cannot use ABRBAC3D without the property of being induced for degradation. So basically, you have to use a wild type virus. For using wild type virus, if you want to detect G2A hypermutation using what I just mentioned, I give you a better picture over here. You may not be able to see anything because in the wild type setting, G2A hypermutation rate is kind of low. So in order to solve this problem, we developed our own assay. We named this next generation sequencing based highly sensitive G2A hypermutation detecting assay. So this is HIV genome. We target to the region which is right after NAF region. The reason we choose this is because this region has a lot of G2A ABRBAC3D G2 hypermutation hotspot. So the routine was we're using PCR to amplify this region. After amplification, the classical method, just like what I just mentioned, we clone this into vector. And after cloning process, molecular cloning process, we isolated the clone and we do mini-prep acylated plasmid DNA, then we send out for Sanger sequencing to analyze each clone sequence. Then we calculate the frequency of G2A hypermutation. This is a traditional method. And at most, you can process like 100. Some lab really process hundreds of clones. You can have like hundreds of arrays. But it's still not enough because in wild type virus, the G2A hypermutation frequency is fairly low. So in our newly developed method, we take advantage of a service called Amplicon Easy service. So the Amplicon Easy service is basically you have a DNA fragment, PCR amplified, you send out to the company. We use a GeneWiz. It's only cost $58. It's very cost efficient. You give them sequence, 1 or 2 weeks later, they give you the result. So they guarantee you have a more than 50K raise. Means more than 50K DNA fragment be analyzed. So this is good enough for a statistic analysis for low frequency mutation. So with this method, we produce 2 viruses. So one virus is we use the 3B. 3B is HIV, one of HIV strain. We infect H9 cells. During chronic infection, we isolated the virus. This is considered virus being produced from T cell. H9 is T cell, tumor T cell line, express HCG. So we expect HCG being incorporated because the cell express HCG. And another cell is the same virus. We infect SUB-T1 cells. SUB-T1 cell does not express HCG, so we expect no HCG being packaged into the virus being produced from this cell. With those 2 viruses, we infect SUB-T1 cell because virus has ABRBAC incorporated in the viral particle. Then we isolate the cDNA, the PCR. We send out for NGS Amplicon Easy sequencing. So this is the frequency mutation we got from this virus, 3B. We call this 3BH9. So those spots were selected only related to ABRBAC3G hotspot, because ABRBAC3G has a hotspot on GG to AG. So we only choose GG, means it's relevant to HCG induced G2A hypermutation. So those let's say in our fragment we have around 200, 199, and this is a spot 36. Spot 36, the frequency is around 6%, and some of them like 16%. So this is the frequency we got from the virus replicated from H9 cells. So versus the 3B virus released from SUB-T1 cells, you see the frequency is much lower. So we at most 1.some%. So so we using this experiment, we argued that the residual amount of ABRBAC3G still has an effect to induce G2A hypermutation in viral cDNA. Okay, so you may want to argue. So H9 from SUB-T1 cell, H9 from 3B virus from H9 cells, 3B virus from SUB-T1 cells. Those virus from 2 different cells. You wanna make sure that the effect was not because of a different cell line. So what we did was we use a transfection assay. So we transfect another viral construct called NF3 with HCG. Basically, in this transfection, ABRBAC3G will be incorporated into the viral particle. The control was the NF43 transfection without HCG. So basically using this one, exactly same cell line. The only difference over here is this one will have HCG incorporated, this one will not. So the G2A hypermutation rate is pretty much similar like what we obtained from the virus produced from T cell line. A little bit low because it really depends on how much HCG you put into your system. So basically, it's significantly reduced if there's no HCG being produced in the cell line, in the viral particle. Then we still saw something over here. If you blew up, use a lower scale, you see that the virus produced from a 293 T cell co-transfected with empty vector or whatever vector, you do not have a HCG. Why you still have some certain extent of a G2A hypermutation happen in those spots? So we wander around and we go ahead and look at because once we did the NGS amplicon easy, we also be able to analyze other mutation other than G2A hypermutation. So other than G2A hypermutation, we saw the similar thing. You see all those, this is most likely spontaneous mutation from RT, right? It's a spontaneous mutation. You see almost all around like 1% or something like that in the same level of this one. So if spontaneous mutation give you some mutation rate like this, you cannot argue this is from HCG because it could be from spontaneous mutation. So that's why we argue our system is sensitive enough to detect spontaneous mutation. So that's why you see the data like over here, you saw some G2A hypermutation happen even without HCG. Okay, so this is from spontaneous mutation. That's what we argue. So with all this, we want to prove this from different angle. Another approach we chose was we want to knock out HCG from H9 cell lines, then produce virus, compare with the virus produced from wild type H9 cell line, then see whether we can obtain the similar result. So in our lab, it's very straightforward to do gene knockout. We have a gene knockout, the knockout rate is 99%. It's very high because we run core, so we're kind of good at this. And the protein expression being confirmed. You see this HCG pattern band, and then after knockout, you do not see. You guys will be able to see my laser pointer, right?
Yes.
Okay. But anyway, so this is after HCG knockout. We move on to do this similar experiment. So this is the virus produced from H9 cells with HCG. H9, the virus produced from H9 HCG knockout cells. So you see the frequency. This one reached up to 2%. This one reached around 1%. So basically, this one significantly higher than this one. And because the virus was produced from H9 cell line, either wild type or knockout, we will be able to do the infectivity assay. So the infectivity assay is we use those two virus infect MAGI cell, we call TZNBL cells. Then we have a readout. We can beta-gal readout. We can reflex back to the virus replication. You see no difference in terms of viral infectivity, even though the cDNA has a difference in their G2A hypermutation rate. So this is a disconnection from cDNA G2A hypermutation to viral infectivity. So another thing move on is we want to show this in another angle. You may I hope you still remember base excision repair is a DNA repair mechanism. So the key component for the reason we mention this is because this repair system will do the opposite to A3G induced G2A hypermutation, because with this system effectively perform its function, it will repair whatever A3G induced C2U mutation. So the key component for this repair enzyme repair system is uracil DNA glycosylase. Basically, it's UNG2. So if you knock out UNG2, you expect G2A hypermutation increase, because this is the key component for base excision repair mechanism. It's doing the opposite way to A3G. So
I'm gonna go talk to a professor in West Basic, but I'll be back in what 40 minutes. I'll be back.
So you been in and out. You sound being cut out. Are you asking a question? Okay. Never mind. So we have a very sufficient knockout. You see the knockout. We did 2 cell line. One is H9, another is 31 cells. So with those 2 cell line, UNG2 being effectively knocked out. And we did a similar experiment as what we did before. So this is in the H9 sub-T1 cell line. This is in the H9 cell line. The blue bar is wild type sub-T1 cell. The orange one is UNG2 knockout. So I want to draw your attention to each spot of the G2A hypermutation frequency. You see almost all of them after UNG2 knockout, the frequency increase a little bit, even though it's not like a huge increase, but all of them increase. The additive effect may be high. So the similar thing happened in the H9 cell lines. Then we after infection, we harvest the bars, and we did the MAGI assay, viral infectivity assay. Again, we did not see significant viral infectivity change. So again, argue even though the cDNA step viral infectivity increased, the GTU hypermutation increase, turns out the viral infectivity did not change. So there's a disconnection. And why there is a disconnection? We further analyzed the viral genome G2A hypermutation frequency. This is the input vars. And so the input vars, we did RT, reverse transcription, then we send out for PCR, then send out for sequencing. This is the frequency for G2A hypermutation in the viral genome. You see small frequency because over the time, the G2A hypermutation accumulated, like what we just presented, the clinical evidence show G2A hypermutation happen. So then we infect sub-T1 cells, and we harvest the vars from that one. And you see, when we did the G2A hypermutation analysis, you see similar amount of G2A hypermutation happen between those 2 vars, even though inside the cell, the cDNA G2A hypermutation increased, but the viral produced has not changed. So that means even though something happened in the cDNA G2A hypermutation frequency, but somehow the vars only select unchanged viral genome to be packaged. So this is the central argument for this study. And there is we analyzed the cDNA for G2A hypermutation frequency. We analyzed the viral genome G2A hypermutation frequency. Between those 2 steps, there are one of the crucial steps is integration. So we further analyze whether the integrated cDNA also increased or not. We did a regular routine allele PCR. So the allele PCR to make sure that what we analyzed is integrated DNA copy. And we have a control over here. The first round of loop PCR, if we did not add a tag enzyme DNA polymerase, there will be no band show up in the second round to make sure that it will be amplified. It truly depends on the first round of PCR is integrated copy. Then we analyze, so this is a cDNA, free cDNA before integration, or even include some integrated. And this is only integrated DNA. You see the G2A hypermutation mutation frequency, not much change, around 12%, 14%, up to 14%. So basically, the selection process did not happen in the integration process. Must be something after the integration. Okay, so this is the model we proposed for the second part of the presentation. So this line presenting for the G2A hypermutation rate, we have a var G2A hypermutation in certain state, and in the cDNA stage, the G2A hypermutation increase, and once the daughter vars come out, somehow the var G2A hypermutation decrease, and along the process, HIV infectivity did not change. So this is the model we propose, and what could be the mechanism? We do not have the data yet. We have a model system. So in terms of the potential mechanism, there's a very interesting data being presented in PNAS 2020. So in terms of the viral packaging, this is a viral packaging signal side. In that paper, they argued the stem loop, the stability of the stem loop is very crucial to the viral genome packaging. This is the wild type virus. You only have one AU classical base pair over here. Those three all non-classical or no hydrogen bond at all. So make this one not very stable. This molecule, this genome molecule is prone for packaging. Means HIV virus particle preferentially packaging this kind of a particle. When they introduce two mutations, CC, to make this loop, stem loop stable, more stable, then they found out this molecule is not being preferred for packaging. So means the stem loop stable, more less chance for the var genome being packaged. And if we look at closely, those two GG is a hotspot for A3G generated G2A hypermutation. If we assume one of the gene G being changed to A, right, because G2A hypermutation, G changed to A will be perfectly paired with U. It will increase the stability of this stem loop. It will reduce possibility based on their theory, right? It will reduce this possibility for this molecule to be packaged. And the same thing happened to this one. If GG changed to AA, this one even more stable. So that argue APOBAC3D has molecular potential to make this one unpreferentially being packaged. So this is what we proposed for our next step. So we want to study whether this is the mechanism to push forward the molecule not being preferentially targeted for packaging by the viral particle. The key point over here is once APOBAC3D target to a certain genome for G2A hypermutation, it usually stay into that molecule, do a multiple run, do a multiple spot of a G2A hypermutation. So it's possible the APOBAC3 gene being targeted to this molecule, change a lot of a spot of the genome region, then move on to this region to change this one. Once been changed, it will not be preferred for packaging by the viral particle. So that's what we argue. So in addition of wave-induced apoB3 degradation, and also being inhibit HIV G2A apoB3 induced the G2A hypermutation, HIV wave may have another mechanism. I think this mechanism is possible because it's in the genome level. It may be wave independent, preferentially not packaging whatever being mutated. So reduce the likelihood for apoB3 generated G2A hypermutation to keep its genome integrity. Another I will use another 2 minutes is another potential mechanism is for HIV transcription. HIV integrated genome transcription, the 3 transcription variants. This is a G1 form, G2 form, and a G3 form. This is the 5' region. And among those 3 regions, the G1 form, you see the chart over here, G1 form is being preferred for G2 for viral packaging. 70% G1 form, 30% G2, less than 1% G3. So remember, GG is hotspot for G2A hypermutation, right? If apoB3 mutate this G, this is not going to be G1 form anymore. So maybe once it's targeted, it also induce the trigger the mechanism is not being preferred for packaging. So we will investigate all this in our future study. I want to end this by acknowledging a group of people. The graduate student Joni Martin and the previous postdoc Yudi Wang, both of them have moved on to their career future, not working in the lab anymore. And with the help from other lab members in our lab, they contribute to the work. And with all my long-term collaborators, CV-, Moto, Chris, and my colleague in Muhammad Medical College, and of course with all those support from our center, RCMI Research Capacity Core, Tennessee CFAR Core, and our department. The funding is from DOD, RCMI program, and the Tennessee CFAR. I want to end over here. Thank you for your attention.